Going to talk today about psychopharmacology. Uh, we're basically going to look at neurotransmitters, drugs, and behavior. So let's get going. It's actually the second time I've done this. The first time I fell asleep. So wish me luck. Here we go. Most drugs affect the brain and behavior by changing synaptic transmission. Axons release a chemical that either excites or inhibits the attached neuron. This is called a neurotransmitter or merely a transmitter. The first transmitters to be discovered were the amine transmitters in 1940. There were three of them: acetylcholine, norepinephrine, and epinephrine, also known as noradrenaline and adrenaline. In the 1950s, the amino acid transmitters were discovered: glutamate, glycine, and gamma-aminobutyric acid. If you think about it, norepinephrine, of course, can raise your feelings, make you feel better. So it wasn't until the 1940s, and actually we didn't have any chemicals, any pharmaceuticals that could raise your norepinephrine level, and we didn't even have a possibility of it until later. So if you're thinking about how you treat somebody with depression or anxiety or schizophrenia, wow, we're kind of stuck at this point. We have we don't really understand how neurotransmitters work. These glutamate, glycine, and gamma-aminobutyric acid, these are mostly inhibitory neurotransmitters. It wasn't until the 1970s that we discovered endorphins. Investigators recognized that short chains of amino acids called peptides also served as transmitters. These included the endogenous opioids. The first one that we discovered was endorphin, and since then we have discovered dynorphin and enkephalin. Also, these short chain amino acids. Of these short chain amino acids, there was oxytocin. Oxytocin tells the brain in women when to deliver their baby. Also tells them that they need to produce milk. Substance P is the P stands for peptide. Substance P tells the brain when you are in pain, and vasopressin, of course, is the antidiuretic hormone. Transmitters must meet the following criteria. The chemical exists in a presynaptic terminal. The transmitter is manufactured in the neuron. The transmitter is released when the neuron fires. Specific receptors exist to receive the transmitter. Adequate amounts of the transmitter will make the neuron fire. Blocking the transmitter stops activity. There are two types of receptors: ionotropic or ion-directed. They respond immediately and allow ions to pass through the receptor when the transmitter is present. And metabotropic, which means directed toward change, it responds slowly by accumulating G proteins to activate other ion channels or other changes in the cell. And I'm going to show you a film or a short video, and this is going to explain how the G proteins work. Let's do this. There we go.
When a ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel, and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
Okay, you saw how slow that was, the whole process. Ionotropic, of course, opens much faster. It just opens the channel, and neurotransmitters come through. Okay. Acetylcholine was the first transmitter that was discovered. Acetylcholine is contained in cholinergic nerve cells. The nucleus basalis and the medial septal nucleus in the basal forebrain are cholinergic cells that project to the hippocampus and amygdala to promote memory. Cells in this area are often destroyed in Alzheimer's disease. The drugs atropine, used to dilate the eyes, and scopolamine, truth serum, are both acetylcholine antagonists. They block the action of the transmitter. The pedunculopontine nucleus and the laterodorsal tegmental nucleus control sleep and are cholinergic cells. There are two main types of acetylcholine receptors, nicotinic and muscarinic. Nicotinic receptors are ionotropic and have excitatory functions. Muscarinic receptors are metabotropic and can be excitatory or inhibitory. There are 5 transmitters classified as monoamines in 2 groups. The catecholamines, norepinephrine, epinephrine, and dopamine, and the indolamines, serotonin and melatonin. Dopamine is one of the most abundant transmitters. It is found all over the brain, but 2 of the more important areas is in the mesostriatal system and the mesolimbocortical system. The mesostriatal system includes the substantia nigra to the striatum, caudate, and putamen. This area of the brain does not actually have very many nerve cells, so what nerves are there can control thousands of synapses. Degeneration of this system can result in Parkinson's disease, tremors, and even paralysis. And so the area is crucial to motor control. This can be caused by aging in select individuals, drug use, or frequent and prolonged head trauma. And of course, this happens to boxers. And there's the putamen, and I don't have an arrow. There's the substantia nigra. Mesolimbocortical system, this area is just in front of the mesostriatal area. This area projects into the limbic system, the amygdala, the hippocampus, the nucleus accumbens. Overstimulation of this area and the dopamine pathways seem to cause schizophrenia. 5 types of dopamine receptors have been identified: D1, D2, D3, D4, and D5. D1 and D5 react in similar manners, but very much different from D2, D3, and D4. Antipsychotic drugs that bind D2 receptor sites relieve the symptoms of schizophrenia. Norepinephrine or noradrenaline is released from 3 different areas in the brainstem. The impact of the neurons producing norepinephrine expands broadly throughout the cerebrum, cerebellum, and spinal cord. This transmitter is believed to affect a broad range of behavioral and physiological functions. Serotonin is classified as an indolamine, a subclassification of monoamines. Large areas of the brain are controlled by serotonin producing neurons. New neurons actually release serotonin. The main grouping is an area of the brainstem called the raphe nuclei. 15 types of serotonin receptors have been identified. Amino acids as transmitters. Some amino acids act as neurotransmitters. Excitatory amino acids. Glutamate is the most common excitatory transmitter, and aspartate. Inhibitory amino acids. Gamma-aminobutyric acid, or GABA, is the most common inhibitory transmitter, and also glycine, which makes glutamate the most numerous excitatory transmitter, and gamma-aminobutyric acid the most common inhibitory transmitter. Glutamate uses both ionotropic receptors and metabotropic receptors. The ionotropic receptors are AMPA receptors. AMPA receptors are able to increase with learning, accelerating, and processing function. Kainate receptors, NMDA receptors are the other two ionotropic receptors. NMDA is involved in information processing. Glutamate has a feature known as excitotoxicity, whereby a neuron that is depolarized due to injury or stroke will die. Glutamate is cleared by astrocytes, and this is, of course, a type of glial cell. GABA receptors can be divided into three types, A, B, and C. GABA receptors contain chloride channels and are ionotropic. GABA-B receptors are metabotropic. GABA-C receptors are ionotropic with chloride channels, but they have a different function than GABA-A receptors. Drugs that block the actions of GABA can cause seizures. Many peptides have a prominent role in the brain as neurotransmitters, opioid peptides, endorphins, the enkephalins, and dynorphin. Substance P, cholecystokinin, neurotensin, neuropeptide Y are found in the gut and the spinal cord. Pituitary hormones, oxytocin, and vasopressin. Neurotransmitters work on just select receptor sites that contain a receptor for the select neurotransmitter, and this is like a lock and key. More neurotransmitters work on more than one receptor. For example, acetylcholine in the muscles excites muscular activity, while in the vagal nerve it inhibits functioning of the heart and the digestive tract. So it acts as an excitatory with muscles, and it acts as an inhibitory neurotransmitter with when it's in the vagal nerve. The neurotransmitters in the body are known as endogenous ligands, internally occurring, endogenous. Man-made drugs or exogenous ligands work because they either act as a neurotransmitter and excite or inhibit the neuron, or they block the action of the transmitter and keep it from affecting the neuron. Some drug molecules bind to the receptor molecules for a long time, while others only for a short time. A drug with a high affinity for a receptor will stay with the receptor longer, so less is needed to be effective. A drug with a low affinity for a receptor will require a higher concentration of the drug to be effective. If both the high and low affinity drug is present, the drug with the high affinity will interfere with the low affinity drug. Agonists do the same job as the transmitter, and antagonists block the job of the transmitter. Some drugs bind with the receptor and make the receptor do the opposite of what they were intended to do. For example, they inhibit to excite or excite to inhibit. These drugs are called inverse agonists. Some drugs bind to the receptor in a place that does not bind the transmitter. When the transmitter binds to the receptor, the drug does not allow the transmitter to activate the neuron. Since the drug does not bind in the transmitter receptor site, it is called a non-competitive antagonist. Drugs that act in a similar manner but activate the neuron are known as non-competitive agonists. The drug Respirine acts as a sedative and as an antihypertensive, inhibits the storage of catecholamine transmitters, dopamine, epinephrine, and norepinephrine in the vesicles. Colchicine, an anti-inflammatory and painkiller used on gout, works because it blocks the maintenance of the microtubules and axonal transport. Caffeine competes with the inhibitor adenosine and thus preventing the inhibitory effect. Calcium channel blockers, verapamil for high blood pressure is an example. They block the calcium channel and thus won't allow the activation of the neuron to release the synaptic vesicles. Amphetamines are calcium channel blockers that inhibit the inhibitory neurons that control the release of catecholamines, especially adrenaline, making an individual feel excited and full of energy. Black widow venom causes the over-release of acetylcholine and eventually causes seizures. Cocaine and amphetamines inhibit reuptake mechanisms prolonging synaptic activity. Selective serotonin reuptake inhibitors, SSRIs, prolong the synaptic activity of the serotonin, which in turn combats depression. This is also how it decreases the sex drive, which is regulated by testosterone in the hypothalamus and controlled by serotonin. Alcohol increases the number of receptors for the inhibitory transmitter GABA. Antipsychotic drugs block dopamine receptor sites. Curare blocks nicotinic acetylcholine receptor sites. Physostigmine is a drug given to Alzheimer's patients to increase memory. It works because it inhibits acetylcholinesterase, the enzyme that breaks down acetylcholine in the synaptic cleft. This allows acetylcholine to stay in the synapse longer, and since acetylcholine activates memory, it leads to an improved memory. Opiates are analgesics. Analgesic is a painkiller. Opiate receptors are found in the limbic and hypothalamic areas of the brain, as well as in the gray matter of the brainstem. Synthetic opiates, such as Demerol, can be injected directly into the spinal cord to cause pain blockage. Endorphins are endogenous opiates. Endorphins are released with physical activity or select physical manipulation such as massage, bathing, or breastfeeding. Endorphins tend to not only block pain, but energize as well. Marijuana is the most widely used legal and illegal drug in the United States. The main active ingredient in marijuana is tetrahydrocannabinol, also known as THC. The human brain actually maintains receptor sites that turn out to be cannabinoid receptors. For a long time, no endogenous ligands could be discovered that shared the cannabinoid receptors, but now we have discovered an anandamide which triggers memory and hunger. THC receptors are found in the hippocampus, cerebellum, substantia nigra, and the cerebral cortex. The receptors reduce sensitivity to pain, reduce sensitivity to temperature, reduce movement, and reduces response to external stimuli. Alcohol works on the nervous system in two ways. Initially, it acts as a stimulant. Increased use produces a longer depressant phase. Prolonged use of alcohol can damage nerve cells. Areas affected by long-term use include the Purkinje cells of the cerebellum, superior frontal cortex, and hippocampal pyramidal cells. And drinker versus a non-drinker, as you can see, it is a depressant. The non-drinker is on the left, and the non-drinker is on the left, and the drinker is on the right. Alcohol activates a GABA receptor, increasing postsynaptic inhibition. Euphoria when drinking may be stimulated by dopamine activation. It activates excitatory glutamate receptors, while it reduces the effects of glutamate and interferes with second messenger systems inside neurons. Anxiolytics, or anxiety loosening, that's what anxiolytic means, are medications that treat anxiety. 8% of American adults suffer from anxiety. The major anxiolytics now in use are benzodiazepines, with the most popular being alprazolam, Xanax, and diazepam, Valium. Benzodiazepines work by enhancing GABA receptors, increasing the effect. Benzodiazepines mirror the reduction of stress produced by allopregnanolone, a hormone derived from progesterone. Allopregnanolone binds to GABA receptors, increasing the effectiveness of GABA. GABA, in turn, inhibits the release of corticotropin-releasing hormone, which triggers the stress response. Alcohol increases the level of allopregnanolone in the brain, creating the calming effect of alcohol. Stimulants are drugs that lead the brain to states of more excitation than inhibition. They have an altering and activating effect. Some act by simulating excitatory neurotransmitters. Some act by blocking normal inhibitory processes, as caffeine does with adenosine. Nicotine. Until the advent of milder cigarettes, nicotine was not widely used. Because of the mildness of cigarette smoke, it is more readily drawn into the lungs and thus is able to cover a large surface of the lungs. The harsher forms of tobacco tend to only come in contact with the mucous membrane. And for this reason, cigarettes have caused a lot more problems than cigars or pipe tobacco in the past. Nicotine activates the acetylcholine receptors known as nicotinic receptors. These receptors are found primarily in the neuromuscular junctions and in the neurons of the autonomic ganglia. Nicotine works on the body by increasing the heart rate, by stimulating the sympathetic nervous system, and by stimulating the release of epinephrine. Cocaine is synthesized from the coca shrub. In their natural and more diluted form, the coca leaves do not produce either an extreme reaction or the possibility of addiction, increased endurance, lower sensitivity to hunger, and a vague sense of well-being. Synthesized into concentrated powder or crystals, crack, the effect is much stronger. As a powder, it is snorted through the nostrils for rapid lung diffusion and thus rapid entry into the bloodstream. Crack, when smoked, enters the bloodstream even faster. Cocaine blocks norepinephrine receptors and allows more of the transmitter to circulate in the brain. Because cocaine is a vasoconstrictor, it increases the blood pressure and induces greater heart activity. This can lead to stroke of the newly initiated or the experienced user. Oddly, cocaine leads to less activity in the frontal lobe, the seat of intellectual function. Even after stopping the use of cocaine, the brain does not return to normal metabolism for several months. Children exposed to cocaine in the womb may suffer from irreversible harm to the development of the brain. Cocaine seems to interfere with the ability to focus attention and with some intellectual functions. Amphetamines and methamphetamines have a similar structure to that of the catecholamine transmitters epinephrine, norepinephrine, and dopamine. The amphetamines cause the release of these transmitters from the synaptic vesicles without a nerve impulse. After the release of the transmitters, the amphetamines enhance the activity by blocking the reuptake of the endogenous transmitter, competing with the endogenous transmitters for the enzyme that inactivates them. Thus, the amphetamines prolong the presence of the endogenous transmitters in the synaptic cleft. Amphetamines in the short term, they affect heightened alertness to a state of euphoria. They ward off boredom. They promote sustained effort with little rest and lowered fatigue. However, quality of work is not enhanced. It increases motivation but not cognitive ability. Amphetamines affect the autonomic nervous system by producing high blood pressure, tremors, dizziness, sweating, rapid breathing, and nausea. Long-term use, you develop tolerance, progressive loss of effectiveness of the drug. This may be due to excitotoxicity, sleeplessness, severe weight loss, general deterioration of mental and physical condition. Extreme use may lead to symptoms similar to those of a paranoid schizophrenic, compulsion, agitated behavior, irrational suspiciousness, observation of those who are chronically show symptoms of brain damage, and of course, back in the old days, amphetamines were widely used. They weren't considered a drug until the 1970s. Of course, I started in medicine in 1971, so we had I saw a lot of people that used a lot of amphetamines, speed freaks they used to call them. These people would accelerate their lives, and then they'd die in their 30s and it wasn't really from old age, but in essence it was. Hallucinogens alter sensory perception. The hallucinogens include LSD, mescaline, and psilocybin. The hallucinations are often visual in effect. LSD, which stands for lysergic acid diethylamide, creates its amazing effects by acting on select serotonin receptors, but researchers are not sure exactly why. The structure of LSD resembles that of serotonin. Phencyclidine, or PCP, was developed as an analgesic and an anesthesia, but created such agitated side effects that its use on humans was stopped. Resurfacing as a veterinary anesthetic, its effects became known on the street, and abuse of the drug followed. PCP can become toxic fairly quickly, with low doses causing combativeness and catatonia, stupor, mania, rigidity, extreme flexibility. Phencyclidine can be found in the powder form, dissolved in water as a pill, or as a dark gummy substance. It is usually smoked by placing it on marijuana or another leafy substance. PCP antagonizes the NMDA receptor and stimulates the release of the transmitter dopamine. Research shows that possibly PCP acts on the prefrontal cortex and produces transient psychosis. Research with animals also shows a degeneration of the hippocampus and cingulate gyrus. One of the newest street hallucinogens is the European rave drug ecstasy, or MDMA. The drug is a combination of amphetamine and hallucinogen and affects the serotonin-producing neurons in the brain. MDMA, while it has a calming effect during use, it may actually be creating a state of excitotoxicity if used frequently. Animal studies have shown marked decreases in serotonin axons after use. This may affect mood and memory. Addiction to nicotine, alcohol, and drugs injures the community by running up medical expenses and causing people to die prematurely. Substance abusers commit crimes and cause traffic accidents. The cost of helping addicts control their addiction and that of controlling drug trafficking is high. Babies born to women using addictive substances impairs brain growth. 20% of all deaths in the United States every year are related to smoking. 100,000 of these are due to lung cancer. Of people over the age of 55, smoking doubles the probability of dementia, especially Alzheimer's disease. After 125,000 deaths a year are associated, about 125,000 deaths a year are associated with alcohol, many resulting from accidents and drunk driving. 70,000 deaths a year are caused by illegal drugs. 8,000 more deaths are caused by drug-associated AIDS. Scientists have been looking for cures of addiction for centuries. Many times, treatments have been far more destructive than the addictions themselves. For example, physicians in the 19th century treated alcohol addiction with laudanum, which is tincture of opium. Tincture means that it's mixed with alcohol. Coca-Cola was used as an alcohol substitute until the cocaine in the beverage was changed to the less addictive caffeine. Since each drug has different effects on the nervous system, it is tricky creating a single model of addiction, but that hasn't stopped anyone from trying. The moral model, in a more moralistic era, people blamed drug abuse on the lack of moral character of the abuser. This model was based on an individual's need to ask for divine intervention to throw off the addiction. The disease model, not responding to the moralists that pointed their fingers at the addiction sufferers, the disease model sees addiction as an affliction that the individual acquires and which requires treatment to cure. The disease model has a large number of adherents because it does not deal with guilt and weakness as the moral model did, but sees the addiction as a chronic problem. The physical dependence model is based on the withdrawal symptoms that an individual may go through after they have used a select drug. Drugs of abuse usually produce pleasurable feelings. Withdrawal symptoms usually are the opposite effects of the effects produced when using the drug. For example, morphine is an analgesic. With intravenous use, it produces an orgasmic rush that leads to a state of serene, drowsy euphoria. Withdrawal symptoms include irritability, tremors, and an elevated heart rate. Goosebumps appear, and the individual goes through episodes of temperature and maladjustments, sweats and chills. Nausea is common. As the individual progresses through withdrawals, the physiological changes that have occurred because of the drug to compensate for the drug affect the individual. Withdrawal symptoms can be relieved by readministering the drug. Within 15 to 20 minutes, the individual has returned to a state of euphoria instead of one of painful agitation. While some drugs have striking withdrawal symptoms, such as opiates, others do not. Cocaine and marijuana, for example, have little to no physical withdrawal symptoms. In these cases, especially with cocaine, the individual becomes psychologically dependent on the drug. The psychological pull is just as strong as the physical need that may be experienced with other substances. The positive reward model came from animal research. Since animals are known to consume addictive substances in nature, researchers sought to discover if an animal could learn to take drugs. Using a simple direct feedback system, they discovered that animals learn to induce effects very rapidly. Researchers found that animals would engage in self-inducement even at levels that did not produce addiction. A wide variety of addictive drugs release dopamine in the nucleus accumbens just as food does in a hungry animal. This dopamine originates in the area of the brain referred to as the ventral tegmental region of the midbrain. Ventral tegmental region of the midbrain. There it is, right. I don't have an arrow. Okay, that's all right. This dopaminergic pathway from the ventral tegmental area to the nucleus accumbens serves as a reward system for many experiences. The addictive powers of the drugs may come through stimulation of this pathway. Using a dopamine agonist reduces the cravings for some drugs. Biological factors: males are more likely to abuse drugs than females. There may also be a genetic predisposition. An alcoholic biological parent makes it more likely that a child will be addictive as well. Personal characteristics: aggressiveness and a tendency to act out emotional states are characteristic of drug abusers, especially in youth. Characteristics that negate abuse: school achievement, high educational goals, and religiousness. Family situation factors related to drug abuse include divorce of parents and having an antisocial sibling. Strong ties to parents protects from drug abuse. Social and community factors: high prevalence of drug use in the community, especially by peer group. It is possible to stop on your own. 90% of ex-smokers and 50% of problem drinkers seem to have quit on their own. Drugs for detoxification, drugs that control the withdrawal symptoms, agonists of drugs that produce common addiction states. Methadone is an alpha opiate receptor agonist that constrains the cravings for heroin. So you can use methadone to get off of heroin. Antagonists to the addictive drug: naltrexone is an antagonist to the opiate receptor, blocking the effect of the drug. Anti-craving medications: substances that affect the desire for the drug. Antidepressants have been used with alcohol withdrawals. Medications that block drug metabolism. Anabuse blocks the metabolism of alcohol, producing instead acetaldehyde, which causes vomiting. So when you drink, if you take Anabuse and you drink alcohol, it will make you throw up. Smoking cigarette. Oh, this has to do with chewing tobacco. The reason I put this in is because up in Montana, a lot of the Native Americans chew tobacco. They dip snuff, actually. So you'll sit in a classroom and, 2/3 of your males in the classroom are dipping snuff and spitting into a bottle, which is pretty nasty. And that's the end of the lecture. So I'll see you guys next week.